I'm Irit rappley. I'm the Chief Scientific Officer of Teal, Omics. It's a pleasure to be here. I was writing some notes as I was listening to the previous two talks. This was a benefit of not being the first speaker. And I've actually heard some themes that I wanted to touch on because I think this is what draws this event together. So we've heard about different types of diseases already, vastly different, right? But one of the common themes I've heard is that we're talking about very large populations of people, um, hundreds of millions, even billions of people affected, very, very large economic impact of these diseases, and yet very few treatment options. Certainly in terms of drugs and medicines, right? But even in terms of resources available for people. Another theme that we heard, and I think was very apparent in this last talk from Gilda and Chris, was that every brain is unique. And although historically a lot of diseases of the brain have been diagnosed based on clinical presentation, like a checklist, right? You have memory issues, you have, whatever, you forget what you have, forget where your keys are, you have gait issues, okay, then it's probably this disease, or you have some other things that you can report your neurologist, and it might be that disease, right? But what we're learning is that the underlying causes of these diseases are often quite different, and not every patient will respond to the same treatment, even if it's approved for a particular disease indication. And so I think all of this together is highlighting, it's a recognition in the field over the past probably decade or so that we really need much more nuanced insight into how the brain works and what causes brain dysfunction, what happens when it doesn't work. And that's where Teal Omics comes in. So I just wanted to kind of put it in context in terms of what we're hearing here. So Teal Omics starts with proteomics. And maybe quick show of hands, who knows what proteomics means? Okay, that's about half. That's awesome. And thank you for not raising your hand if you aren't sure. That's also really helpful. So proteomics, we will get to in a moment, actually. I won't go into too much, but maybe very briefly, Teal Omics was founded out of Stanford from the lab of Tony Weeks-Correa. He's been one of the foremost researchers on aging, and specifically aging of the brain, for the past 20 plus years. He's done a lot of amazing things in his career, and one of the questions he has been focused on asking is, what happens during aging? What is different in an 80-year-old brain or an 80-year-old body than a 60-year-old body than a 20-year-old body? So one of the foundational insights coming out of not just Tony's lab, but one of the foundational insights for the work that we do is that blood connects to all organs. And we tend to think about, we most of us probably learned in school that the brain is a privileged organ and nothing gets past the blood-brain barrier and all of that, right? But the brain still needs glucose and oxygen. There's lots of blood that flows to the brain. In fact, uses up about 25% of your glucose and oxygen, even though it's only about 10% of your body. The blood connects all organs. And conversely, and this is something that's being recognized more and more recently, all the organs secrete things into your blood. So kind of detritus from organ function, metabolic debris, but also functional molecules from every organ are circulating in the blood. So when we collect blood, we can determine, we can assess what is in it. And the science of these large-scale unbiased assessments is just kind of seeing everything that's in there. These are omics. And if you're looking at genes, you're doing genomics. And if you're looking at proteins, you're doing proteomics. And if you're looking at fat, you're doing lipidomics. And if you're looking at metabolites, you're doing metabolomics. And people are inventing new types of omics almost every day. So at Teal, we're focusing on proteomics. So we're starting by identifying the different proteins that are circulating in your blood. So just by taking a regular blood draw, there are instruments available that can identify thousands of proteins that are in your blood. These proteins come from all over the body. Some of them come from your immune system. Some of them come from your skin. Some of them come from your liver. Some of them come from your brain. And thanks to mapping, extensive mapping that has been done over decades, we can identify which proteins came from which organs, because we know where those proteins tend to be expressed most highly. So when we do that, we create signatures, like fingerprints, for every organ based on the protein that it secretes into the blood. We can follow how these fingerprints change over time. So we have looked at thousands, tens of thousands of blood samples from different people at different ages. And starting only with healthy people, we've created an understanding of what does the brain signature, for example, look like for a 20-year-old person versus a 30-year-old versus a 40-year-old, etc. And we've looked at what does the brain signature look like for somebody with Alzheimer's disease versus somebody with Parkinson's disease versus somebody with diabetes versus somebody with chronic kidney disease, etc. So we start by creating these signatures, and we can compare the calculated age of an organ, let's say the brain, to the chronological age of the person at the time that they donated the blood. If I were to go and donate blood and have my organ signatures done, I might find that my brain signature is 5 years older than the rest of my body signature, or than my chronological age. That might be an indicator that my brain is going through some process, right, some aging process faster than the rest of my body, or than we would have expected. Conversely, I might find that my brain signature is 5 years younger. That would make me. So we can look at these organ-specific signatures and look at organ-specific ages. We can also calculate, because we're looking at all of the proteins in the blood at the same time. What you see on the left side, we can also calculate an organ-wide, an organismal aging signature. There's a lot of information in that too. As a whole being, are you aging faster than the number of years on Earth would have suggested that you should? And we call these age gaps. And it can be positive if your organismal signature is older than your chronological age. So they can be negative if your organs appear younger than we would have predicted. And based on these signatures, we can learn a lot about the underlying disease processes that are going on. So we start again on the left. This is just a different version of what I showed you a couple of slides ago. Blood connects all organs. We can start by looking at proteins that are circulating in your blood and assessing the fingerprints of all of the different organs. We can look at how those organs change as a whole. This is the organismal signature. And what you see in the middle panel is a panel from a paper that was actually published several years ago by one of our advisors, showing that that aging is not a linear process, but there are, in fact, two peaks when aging processes happen more. One at around a little before the age of 40, and another one around the age of 80. And this information, this was actually recently repeated in a big headline and a lot of news. You might have seen it a few months ago, but this figure that I'm showing is actually from a paper several years ago that had already. So that's looking at the organismal, the whole organ signature, right? But we can also look on the right side, you see, at organ-specific signatures and correlate what is happening to a particular organ in any given disease. And what we find is that sometimes it makes a lot of sense, right? Like chronic kidney disease is associated with changes in the kidney signature, as you would expect, right? But we also see that, for example, Alzheimer's disease is associated with changes in the arteries. And this is not super surprising because there have been studies suggesting and linking vascular changes to Alzheimer's disease for many years now. It's very interesting that we see it just by looking at all the proteins that are circulating in the blood. So this work was published from Tony's lab at the end of 2023. And on the left-hand side, I'm showing the title of that paper and some of the key figures from it, describing these organ signatures of aging in blood. Other groups are continuing to work on it, expand on it. There's more research being done every month. Some of it has been published, some on the medRxiv and bioRxiv, these preprint servers. Some of it, like the Irit work in the middle, is work that we're doing in collaboration with a group at the NIH. But people are following up on this idea, gaining a lot of traction to look at proteins in the blood and try to use them to understand aging and disease processes. And that's what Teal is. So what does it actually look like? What information can we actually get? Here I'm showing two different versions of the same graph. And this is really important because we have to be able to show that different people can get the same or largely the same information, largely the same result when they do the work in different places, right? So on the left-hand side is a figure from a preprint from Tony's lab that was done after Teal was founded. So this is work that we are no longer directly related to. And then on the right is the work that we did internally to try to ask the same questions and see if we got the same. And what you can see is that overall, largely, yes, we got very similar answers. I want to point out really interesting insight that, well, first, actually, sorry, I look at these graphs all the time, but you don't. So let me explain to you how to read graph, what you're looking at. We're looking, so the graph is representing Alzheimer's disease risk and the influence on Alzheimer's disease risk of a change in the brain signature or in the organ signature. So along the y-axis, along the vertical axis, you have the different organ signatures, including also a conventional signature and an organismal signature. A conventional signature just means we took all of the proteins as one big soup and used that to calculate the signature or the gap, right? And the organismal signature means we took the sum of all the different organs and looked at it that way, just slightly different variables. Along the x-axis, you're looking at a hazard ratio, which means what is the increased risk associated with a change of a particular magnitude in that organ signature. So if we look at the very top, brain, you can see that the brain age gap, so having an older brain, is associated with approximately a twofold increase in your risk of getting Alzheimer's. And it's approximately twofold in both of these graphs. It's not exactly twofold, but when you're looking at this type of data, what you're seeing here is a very, very good correlation. Okay, so it's not surprising that the brain signature has the most impact on your risk of Alzheimer's disease. I mean, I could have asked you which organ you think would have the biggest impact, and everybody here would have said the brain. That part is it's not surprising. It is surprising, to me at least, that the brain signature alone is more impactful than the whole body signature, right? Because again, the proteins are coming from all over your body, your blood is circulating. Another thing that we've found, so we can do this for any disease, and we can find similar risk for any disease, not just neurological disease. Teal was founded to look at CNS diseases and diseases of the immune system. But in principle, we could do this for diabetes, we could do this for heart disease, we could do this for any disease that we want. Now, proteins, one of the reasons people haven't looked at proteins so much in drug discovery until very recently, I think there are two big driving reasons. One is that we didn't have the technology to identify thousands of proteins at a time. And that was all maybe 10 years ago, when it's the scale that we need and with the type of samples that we work with from blood. But another one is that in general, genes are seen as the driving factors underlying disease. And we always talk about genes. You heard Robert talk about MECP2, right? We know that there are genes associated with Alzheimer's disease. We've all heard of ApoE. We've all heard of A beta, right? There are genes associated with most diseases today. But the genes don't tell the whole story because we are born with those genetic mutations. But these are diseases that only develop often when we're 67 or 80 years old. What happened in that interim that led to that disease? At Teal, we believe that the proteins will tell that story. But we do acknowledge that genes help to tie it back to causation. How do you know that what you're looking at is causing the disease and not a manifestation of the disease or something that happened that's unrelated? So we look at genetics, and what this graph is showing is a genetic causality analysis that's called a GWAS, or right, and you've probably all seen some version of this or heard some version of this. It's a genome-wide association study. So across the x-axis, we're looking at all of the chromosomes and essentially all of the genes. So every chromosome has a different color, and then every dot is really a different gene on that chromosome. And on the y-axis, you're looking at the fold change. How much of a change, how big of a change are we seeing in that gene with a particular single nucleotide change? One single nucleotide base of change. It's something that's known, identified in a sample, and it's different from the typical, right? So it's divergent in that sense from the typical genetic code. And what we look for is, these are called Manhattan plots. I love that because they look like skyscrapers. So what we're looking for is the tallest skyscraper, right? And the dotted red line along the bottom is the cutoff, is what's considered like baseline, not a big difference. And we're looking for the things that are really tall skyscrapers in these Manhattan plots. And then those are, because of the way that these calculations are done, those dots at the tops of these skyscrapers are considered to be causing the effect, the thing you're looking at. And in this case, it's Alzheimer's. So when we see a dot that represents a gene that codes for the protein we identified in the blood, we've made that connection. And we've said, we see this protein change in blood, and we see this dot in the genome, right? And that the gene is different. And that is telling us there's probably a causal effect. So, other cool things, other reasons that we believe this platform is the way that we will get to the nuance that we need in order to fully understand diseases of the brain. I want to focus actually on the farthest right graph. So that graph is showing the brain age gap, and that's what we're looking at on the y-axis. The x-axis is time in years, and 0 represents the time when someone was diagnosed with dementia. So this is not Alzheimer's disease, it's any type of dementia. So to the left of 0, the negative numbers are time before dementia, right? Before a diagnosis of dementia. So these are people who came and donated blood and they were perfectly healthy when they donated. And then at some later point, they were diagnosed with dementia. And what you can see is that up to 20 years before they were diagnosed with dementia, their brain gap was elevated, okay? Now, I'm not suggesting that here, boom, we have a diagnostic and everybody should go get their brain age gap measured, right? Because these are not huge differences. We don't know exactly how to identify what would count as a positive signal. But what I am saying is that there's clearly something going on in the brain that we can see in proteins and circulating in the blood 20 years before a diagnosis of dementia, right? There's some change happening in the brain. When someone gets diagnosed with dementia, it didn't start yesterday. It started 20 years ago. And so if we could identify what in that signature is driving that change, what is driving those processes, we would have much better targets for treating dementia or autism or Rett syndrome or whatever other disease of the brain we're looking at. And what we know is this is not going to be the same for everybody. Every brain is different. But because we're looking at signatures, we're looking at signatures, as you can see in the middle graph, that are composed of many different proteins, not just one or two. We get the nuance that we can look and say, oh, this one protein, your TFAP is kind of within the normal range, but maybe your NEFL is not. We can start to get that nuance that is missing when you only look at one specific readout. So we believe that this is kind of the future, essentially, of neurodegenerative disease research. We're not the only ones to say this. This graph is intended to kind of show you what we already kind of know, that just like we no longer talk about cancer as only as breast cancer versus pancreatic cancer, we know that there are different subtypes that are based on the genetic makeup. Similarly, in the near future, we'll talk not about Alzheimer's disease or Parkinson's disease, but about these molecular subtypes. And this is just a retelling of essentially what I just told you. I'm happy to come back to it if you guys have specific questions, but I want to be mindful of time and I want to leave time for questions. So I'll just end with this to say, okay, like I've told you about these signatures in the brain and in the blood and brain signatures and other organ features, but like, how do we put it all together? And this slide kind of lays out the funnel that we have built in the platform that Teal is working. So again, we start with aging. We look at all the proteins in the blood and we see how are they changing with age in these different signatures. Then we look at, are there specific organ signatures that are changing, and are there specific disease signatures that we can identify? We also, and I didn't touch on this very much for this talk, but I'm happy to go into more detail if you're interested. We also look at the network analyses, the networks that all of these proteins are part of. Because one protein might be secreted into the blood, but that protein never acts alone inside the cell. It acts in a pathway. It has partners. It does things to other proteins. And so we look at all of those interactions to see whether there's any useful information there about the processes that are happening in the course of a disease. Again, I showed you those Manhattan plots. We look at genetic causality. We try to correlate the protein changes with genetic changes. Because it is really important to know that we're going to target the cause of a disease and not just the symptom. And we look at cell types, can we tell which different cell in the brain, what region in the brain, et cetera. And of course, we always look in the context of what other people know and what else has been done, although we are looking for novel targets. That's what Teal is up to. And I think that's it. I'll take questions.
Great presentation. So do I understand correctly that your platform allows to evaluate chronological age versus biological age, and then from this gap, you can assume or say that the risk for dementia is higher, right?
Potentially, yes.
But if you're working in first of all, your graph shows that it's a linear correlation, but I assume it should be exponential, because when you're born, it's 0, 0. When you're 1 year old, it should be, and then at some point it starts diverging. So I'm not sure that using linear approximation would be the best choice, maybe at older ages. And second question is, we know that to get protein in the brain, you have to fight blood-brain barrier. I mean, to get protein out of the brain, I assume you also have to fight blood-brain barrier. It means you have selection of proteins that might be secreted from brain into the plasma, which bias a little bit your panel. If you know that you work with brain, will it be easier instead of, I know that blood is easy to take and was sensational, but wouldn't it be easier for you to take CSF and look at protein there, because it would be give you more accurate information?
Yeah, those are excellent questions. So to your last question about blood versus CSF, for the brain, potentially CSF is a more proximal measure, right? A tissue that's closer to the brain. And you're right that there isn't, that it's on the other side of the blood-brain barrier, for the brain side of the blood-brain barrier. So you're not fighting against that. And you will probably see a different signature. You'll probably see different proteins in the CNS than you do, or in the CSF than you do in the blood. Is that necessarily better? I don't know. We'd have to do that study. If I asked you, would you rather go in for a routine blood draw or a spinal tap? But if you don't know, I agree with you. I'd rather have an exact diagnosis too. Yeah, yeah, I understand. So just to repeat for people online, the comment was we're biasing ourselves by looking in the plasma, in the blood, to only those proteins that have crossed the blood-brain barrier. We're leaving behind some things that we would be able to find in the CSF. It's absolutely true. Our hypothesis is that there is sufficient information in the blood to be able to make useful statements about the processes underlying disease causality and early disease processes. One benefit of working in the blood that you would lose if we were looking in the CSF is that the blood contains information from organs other than the brain. And so we could create an Alzheimer's signature. In fact, we are working to create an Alzheimer's signature that is looking at changes not just in the brain, but also in the arteries and potentially, in the liver and the kidney or in any other organ where we see important changes. We don't have to be able to say why those are there or why they might be meaningful for Alzheimer's disease right now. That's the work that's going to happen over the next 5 to 10 years to understand why those changes are happening. But we are losing some information by looking in the blood rather than the CSF, but we're gaining a lot of other information, which we believe by looking at the person holistically at all of the organs will be more beneficial in the long run than looking just at the proteins in the CSF. But it is a hypothesis. It is one approach. It is absolutely not the only and probably not the best approach. I think it's just going to take time. I would love to see someone else do the same thing and build a similar platform in CSF. We have just decided to start with blood. But it's a really good point.
We have an online question.
Thanks, Irit, for that presentation. Yeah, there's a question online here, and you might have addressed it a little bit in your previous answer, but could you talk a little bit about how GWAS, how that aspect addresses causality?
Ooh, yes. I can talk for a really long time about that, but I'm going to try not to spare all of you. So GWAS are, again, genome-wide association studies. So they're looking at a single base change in a gene, right? So genes are made up of letters, essentially. That's how we represent them, right? A, T, G, C. And if you change one of those, that's a mutation, but we call them SNPs, single nucleotide polymorphisms, because most of them don't really have a functional effect that we can tell. So most of us are walking around with thousands of these single nucleotide polymorphisms in our various genes with no apparent detriment, or none that we know how to measure. There's an agreed upon sequence to every gene, and then if your sequence happens to be a little bit different at one point, you have a SNP. So we look at these. Most of these, some few of these have been shown to actually cause a disease or to cause very high elevated risk for a disease with some high level of penetrance. But the majority of them are kind of not known to do anything functional, at least not that we can tell clinically. But we can see that some of these are associated with higher or lower levels of the protein being expressed. So every gene codes for a protein, right? And some of these SNPs affect the protein level. If we are measuring changes in protein levels as part of a signature, and if we see a corresponding SNP in the gene that codes for that protein, that's known to lead to elevated or decreased levels of that protein, right? Because a lot of these SNPs do. Now we can say, oh, there's a genetic cause for the change protein level, the difference from your baseline. And again, because the genes are there all along, we're born with these SNPs in our genes. The genes are the closest thing we have in biology of disease to causality. It's the best kind of smoking gun we have to say, okay, if this is already in your genes and you're getting this disease, it's a good chance that's what's causing. That's what.
Yeah, thank you. Another follow-up question to that. So outside of the genome, does diet or environment affect the aging profile?
Yes, it probably does.
Uh-huh.
And one of our challenges right now is that for a lot of the samples we're working with, we don't have the exact history of like what the person ate for breakfast before they donated blood, for example. Right. And so there's some amount of noise in our system that we have to work around for now and try to find signals that are higher than that. In the future, I would love to do a study like that and say what happens if you drink a cup of coffee, how does it change your organ signatures versus if you eat egg McMuffin, what does that do, right? That is a much farther down the line work, but it's one of the reasons that we have chosen to focus on proteins rather than genes, because they are responsive to our environment, to what we eat, to stress, to disease processes in real time. And so we believe we can identify disease processes then by the changes that are responsive.
Thank you.
Quick question. The UK Biobank had produced a large number of GWAS hits as well, using exactly the same approach. Are your approach or results similar to what they have got?
Biobank data as part of our of the data set that we're curating. So we're collecting data from many different data sets. Some of them are publicly available, like UK Biobank. Some of them only through our collaborators, like Irit. And we're putting them all together into our platform. So yes, we have looked at that. It is the UK Biobank provides a lot of the data that we're based on.
Any other questions? Okay, thank you very much.
Thank you.